Hello, ninth standard. Have a good day. I hope you have gone through the previous class lesson. I am sure you are able to follow that also. Right? Today we will continue video number 4, the same tissues lesson. It will go for another 2 more classes. Right? And before that, I would like to give an information that you need to prepare enrichment activity. As you know, it can be a model or a chart or any improvised material you need to do, right? And this time we are asked to do for every lesson. For 9th standard, we have to simple activity only. We have to prepare every lesson, and from that, best of 2 marks will be awarded for internal assessment. Right? And I hope you know that your 9th internal marks will be sent to the board. I hope you are aware of it. School will also give you the information. Okay. And now for first lesson you have learned fundamental unit of life, that is cell structure. Is it not? For that cell structure, prepare a plant cell model. Okay, prepare a plant cell model using the items on the pulses variety. And it is also said we have to that CBSE board has done some combination that we should always know about details of other states also. So I would like to have about Jammu Kashmir content, as we have last year 8th standard we have learnt kharif crop and rabi crop. In that Jammu Kashmir they are mostly as seasonal climate, so they will have winter and also summer peak they will have, whereas Tamil Nadu will have moderate climate. Is it not? So on that, this kharif crop and rabi crop, whenever with the examples of winter season crop, that we will use it for plant cell outline and also the content. The diagram you will refer. Last year I told you plant cell notes you should not throw it off. Is it not? And also this year for revision, plant cell PPT presentation I have given. With that you get a model, plant cell model, that is ultrastructure of a plant cell. That model you have to prepare. And same way animal cell. So for Tamil Nadu, using Tamil Nadu pulses you prepare for plant cell. For animal cell, Jammu Kashmir என்னென்ன crops விளையுமோ அதோட examples இங்கே available. ராஜ்மா, வீட்லயே different types அந்த மாதிரி இதை outline எடுத்து வச்சுப்போம். Comparison of rabi crop and kharif crop ஆச்சு நம்ம டயக்ராமும் மாடலும் ரெடி பண்ணாச்சு. Okay, that is for fundamental unit of life lesson. I hope you are able to follow this. Sure. Then we will continue this lesson. Another I will give this tips you need to make use of it. Okay. So yesterday we have been learning plant tissues, classification as meristematic tissue and permanent tissues. Meristematic tissues are present in the actively dividing region as root tip, shoot apex and flower bud. I hope you remember this. And meristems are of 3 types: apical meristem, intercalary meristem and lateral meristem. I hope you understand this. Sure. And then this meristematic tissue, they are helpful in increasing the length of the plant, increasing the height of the plant, increasing the girth of the stem portion. That is for the activity or function of meristematic activity cells. Okay, tissues. And these meristems, they have characteristic feature as prominent nuclei, absence of vacuole and cytoplasm will be present, and cell wall is very thin in nature. I hope you can recollect this, right? Then permanent tissues, as I said, it is of parenchyma, collenchyma and sclerenchyma. Parenchyma gets different names as with the chloroplast, chlorenchyma, with the air spaces, parenchyma, and with the storage material, it is storage parenchyma. In the center of the vascular bundle in the stem, it is pith. In the center of the vascular bundle in the root, it is known as conjunctive tissue. Then collenchyma and parenchyma, the cell wall material is cellulose and hemicellulose. Whereas collenchyma, when this parenchyma matures, by the addition of cell wall thickening pectin, it becomes collenchyma. And that collenchyma, that cell wall material will be confined to the corners only. Confined to the corners. இது வந்து collenchyma tissue இந்த மாதிரி கார்னர்ல தான் deposition of pectin material. சரியா? And then actually it is a living tissue and it is flexible in nature. Collenchyma tissues are flexible in nature. And third type is sclerenchyma, where it is almost a dead protoplast by the addition of or by the deposition of lignin. The entire sclerenchyma tissue will have hard and thick lignified wall. Lignin is present. Hard, thick and lignified wall. And this sclerenchyma, it is of two types as sclerites and fibers. Fibers will be present in the husk of coconut. The coconut tenna chowdi solluvala, tenna mattai. That is husk of coconut. That is full of sclerenchyma fibers. I hope you understand. And the sclerites, they are also known as stone cells. They are having arranged in a lamellar structure. That is stone cell. And these stone cells, they are present in the fruit wall of pear and nuts. So that as it is giving a gritty nature. I hope you understand. Now this diagram, what is drawn in the blackboard, it is your whiteboard, it is your textbook diagram. In the textbook diagram, we have first parenchyma. This is transverse section. That is, when we have this as a stem portion, we cut like this. Appo the surface like in the surface la tissues irukum. That is transverse section. Whereas this is LS, the same stem portion, when you cut this way vertically, then it is longitudinal section. So this is LS na longitudinal section, TS na transverse section. Right? So here parenchyma, it has intercellular spaces. Same way here it has intercellular spaces. Right? Whereas collenchyma, it is again in our textbook, it is this is transverse section and this is LS longitudinal section. This is transverse section, this one longitudinal section. So this single or a group of, suppose when we talk about this parenchyma tissue, these tissues only becoming collenchyma by the addition of pectin. These collenchyma only will become sclerenchyma by the addition of lignin. So separator adengilla molecule. Idhe parenchyma da collenchyma va marum, sclerenchyma va marum. I hope you understand. So this is so for everything in our textbook we have transverse section and longitudinal section diagram. So here in collenchyma, idella lines, The thickening is confined to the corner of the cell. Confined corner in the confined to the corner of the cell. That is the diagram given in a textbook. I hope you are able to follow this. The next one, it is these now these tissues, it is the diagram. All these diagram what I have drawn in the board as transverse section, longitudinal section. And then this paragraph is about the function of Sclerenchyma tissue. Husk of coconut made up of sclerenchymatous tissues. The cells are dead and they are long and narrow as the walls are thickened due to lignin, which is a chemical substance which acts as a cement and hardens them. Right? Then this tissue is present in stem around the vascular bundle in the veins of the leaf and a hard covering of seeds and nuts. The location where are they present, they should know. I hope you understand. Then this one again, for example, this tissues again, what we are talking about, we have, say for example again, I am taking a transverse section of stem. Now this transverse section of stem will be like this. Okay, and then inner to that, it will be arranged like this. It will be arranged like this. This portion is the vascular bundle and this is the epidermal layer. Otherwise we say this is epidermis. Now here, when we just take this portion as enlarged, it will have the structure like this. Epidermis will be drawn like this. Okay, so this is the outermost layer, epidermis. Now this epidermal layer is formed of compactly arranged rectangular cells. Epidermal layer, they are compactly arranged rectangular cells. Okay, at the same time, this epidermis will bear certain structures meant for certain functions. For example, if we take root, when we take root, the epidermis of the root will be having structure like this. Will be having structure like this. This is root hair. The root hair is meant for absorption of water and minerals from the soil. So this epidermis of root will be a root hair for absorption of water and minerals. Okay. At the same time, in root, the epidermis, it is called epiblema or rhizodermis. It is called epiblema or rhizodermis because the outer portion of the epiblema is mixed with the soil. So the soil, it will not have a smooth surface. So that ruptured, broken epidermal layer will be seen along with the root hair. So it is called epiblema or rhizodermis. So that is for the root hair. The same way, this epidermis in the stem and leaf, it will have a structure called stomata. Singular stoma, plural stomata. The stomata structure, it will be having a bean-shaped cell like this. Structure of stomata, bean-shaped cell called a guard cell. And this guard cell will have nucleus and numerous chloroplasts. God cells will have nucleus and numerous chloroplasts. And these are the cells surrounding the God cell, epidermal cell. These are surrounding cells. In the epidermis, these are epidermal cells. Okay. So the exchange of gases is taking place through the stomata. Through the opening and closing of the God cells, exchange of gases will occur. What are the gases to be exchanged for? For photosynthesis, carbon dioxide is taken in and oxygen is given out. For respiration, oxygen is taken in and carbon dioxide is given out. Same time, the oxygen released during photosynthesis can also be utilized by the green plants for respiration, and that is called photorespiration. Because normally photosynthesis, light reaction will occur only during daytime. So that oxygen is released only during daytime, and that oxygen is taken by the green plants for respiration. So it is called photorespiration. I hope you understand. So the epidermal cells are in the paragraph. Already the video is having a clear content. Only thing I am just recalling the textbook content. So here this is about the Sclerenchyma structure and function, and then it is about the God cell, epidermis. It will be usually made of single layer of cell, but in some plants, especially in desert plant, it will be covered by a layer called cutin. Normally, green plants will have epidermis like this, right? And it will be covered by another thick layer called cuticle. Now this cuticle is having the deposition of cell wall material called cutin. And in desert plant, during hot summer when water is not available, this presence of cuticle will prevent transpiration. What is transpiration? Evaporation of excess water from the leaves. That will be prevented. So that cuticle, suppose if it is releasing, so if it excess water vapor is there, that is called cuticular transpiration. Normally in desert plant, it is highly protecting. The cuticular layer in the epidermis is highly protecting the stem and leaf by avoiding transpiration. And at the same time, this epidermis in the stem will also have epidermal hair. இங்க வந்து நம்ம root hair சொல்றோம்ல, அது மாதிரி here we can say shoot hair, but we should say it as epidermal hair. I hope you understand. And then in some tendril and climbers and the soft stemler, the stem and the velvet marry hair-like projections. There are epidermal projections. Okay, so this is for the epidermis that is given in this paragraph. First about the epidermis, then its function. As the entire surface of the plant has outer covering epidermis, it protects all parts of the plant. The epidermal cells on the aerial parts of the plant often secrete waxy, water-resistant layer on their surface. That is cuticle. It is covering the layer. This aids protection against loss of water, mechanical injury, and invasion of parasitic fungi. It is the function. And then this paragraph, structure of stomata. Then this paragraph explains about the presence of root hair in the root epidermis. Root epidermis it is epiblema or rhizodermis. Right? Then this is about the cuticle. I hope you understand. Then this paragraph, last paragraph, in page 72, it says about the structure called cark. The bark of the stem, portion, the bark of the tree trunk, the bark of the stem portion that increases the thickness, that increases the thickness of the stem. That is done by the bark region where it contains structure called cambium. Cambium will also show meristematic activity which is present inside the vascular bundle of the dicot stem. Okay, and that increases. It ஒரு பெரிய tree trunk பண்ணி கட் பண்ணி போட்டிருந்தோம் அந்த மரக்கடையில, it has rings, brown rings, 2 or 3 இருக்கும் இல்லையா, அதெல்லாம் showing the year, number of years the plant, the trees, how old are they, the number of tree, the year சொல்லும். So ஒரு layer பேரு annual ring, and ஒரு year that increase in thickness. இந்த diagram, this portion is called cambium. இது ஒரு vascular bundle structure enlarged. This portion cambium will helping in secondary thickening. We'll just continue. Yes, we'll continue. This page 72. Please understand. Sclerenchyma, I repeat, sclerenchyma, and then it is about the epidermis. Epidermis, we should learn about epiblema or rhizodermis for roots, and it gives rise to root hairs. And then in stem, it is just epidermis, compactly arranged rectangular cells, and it may give rise to shoot hair or epidermal hair. And then this is about the function of epidermis. Note CBSE, easy to learn, right? And then this paragraph is about the structure of stomata, structure and function of stomata. Stomata main function, exchange of gases and transpiration. The process of transpiration will occur. Exchange of gases for photosynthesis and respiration. So this paragraph about stomata, right? There is one question. Think about which gas may be required for photosynthesis. Find out the role of transpiration in plants. The hard question again. Think about the gas for photosynthesis. Carbon dioxide is taken in, oxygen is given out. For respiration, oxygen is taken in and carbon dioxide is given out. As I said just above, earlier, the oxygen released during photosynthesis can also be taken by green plants for respiration, and it is known as photorespiration because the released oxygen is taking place, released oxygen during light reaction that occurs only in presence of sunlight. So it is called photorespiration. And then find out the role of transpiration in plant. Excess evaporation of excess water from the green plant. And then transpiration is a necessary evil. That you have to, 3 points you have to learn. Okay, and then this paragraph, root hair. Then about the cuticle. Cuticle will protect the epidermis by avoiding transpiration, especially in desert plants. And then next one. Is the outer layer of branch of a tree different from outer layer of young stem? Yes, because young stem will have soft and tender nature but very little harder, Whereas as the plants grow older, the outer protective tissue undergoes certain changes. A strip of secondary meristem replaces the epidermis of the stem. So the cells on the outside are cut off from this layer, and this forms the several layer of thick cork or bark of the tree, and that is done by the secondary meristem, that is cambium, present inside the vascular bundle. So like that we have the points correct note பண்ணிட்டு you have to carry on. So this is for the simple permanent tissues, right? Now we will learn about complex permanent tissues. Complex permanent tissues are xylem and phloem. Complex permanent tissues. Complex permanent tissues are xylem and phloem. Xylem and phloem. Now the xylem, it conducts water and minerals. Conducts water and minerals. Phloem conducts food material. Xylem conducts water and phloem conducts food materials. Yeah, that we learned already. Now here, the xylem, we see complex permanent tissue because the xylem, it is formed of xylem tracheids, xylem vessels, and xylem parenchyma and xylem fibers. Xylem parenchyma and xylem fibers. Normally, this xylem and phloem, they are arising from the apical meristem region. Suppose this part of it, I say a group of cells like this. A group of cells from this apical meristem region is differentiating into xylem. What happens? This group of cells, for example, ஒரு 9 tissue cells வந்துருக்கு. In a group of tissue 9 cells இருக்கு. Now these tissues, they are differentiating into xylem elements or xylem components. They are xylem tracheids, xylem vessels, xylem parenchyma and xylem fibers. அதனால the 4 different components or elements இருக்கிறதுனால it is called complex permanent tissues. சரியா? Now this xylem tracheids and vessel, only thing, tracheids are long, narrow and elongated pipeline structure, whereas vessels, they are broad, elongated pipeline structure. Only thing, the width, it is very narrow, this is wide. So this is xylem vessel. This is xylem trachea. That's the okay. So xylem tracheids and vessel, they are very long pipeline-like structure with the xylem thickenings. Xylem thickening, they are nothing but cell wall materials. So now this xylem tracheids and vessels, say for example, in a multi-story building, from the ground floor to the top floor, the water connection is there. Yeah? So similar to that pipeline structure, it is as tracheids and vessels. Tracheids will be narrow. இப்போ நம்ம வீட்டுக்குள்ள பைப்லைன் கனெக்ஷன்னாக்க ஜஸ்ட் அரை இன்ச் பைப்பு, வேற மழை தண்ணி வருதுன்னா அது வந்து ஒரு பெரிய இதாக இருக்குது, பெரிய டியூப் பிவிசி பைப்பு. அதே மாதிரி தான் இது வந்து 1 இன்ச்சு, இது 5 இன்ச்சு. ஸோ திஸ் இஸ் நேரோ, திஸ் இஸ் வைடர். And the cell wall, it is made up of cell wall thickenings. Okay. Now this cell, so this is what tracheates and vessel. So this 9 tissues there. Suppose 2 cells are forming tracheates, 3 cells are forming vessels. So on the cell at the, so mean the 9 of 4 they are remaining as xylem parenchyma tissue. So now I say these 9 cells, they are parenchyma tissue, xylem parenchyma tissue, that it gets differentiated into tracheates and vessels, right? And then later when this parenchyma becomes mature, it is called fibers, xylem fibers. Now this xylem fiber is called a wood fiber. Xylem fiber is called wood fiber. I hope you understand. Yeah? So now this xylem, we say it is getting thickening. Now this cell wall thickening in xylem. Suppose I am drawing this as trachea. Okay. This trachea, when the thickening is arranged like this, this is called annular. Annulation ring like that is annulation. So this is annular thickening. Okay. When it is in spiral form, the spiral shape thickening now cell wall material will be deposited. This is spiral thickening. Then when it is ladder form, it is scalariform thickening. Ladder form and the scalariform thickening. Right? And then in some arrangement, when it is in some region, it will be having pits like this. This is the cell wall thickening. So this arrangement of thickening of cell wall material in xylem, it gets different name. Annular ring-like structure, annular thickening, spiral thickening, ladder shape, ladder shape, that is scalariform, and this is called pitted thickening. This is known as pitted thickening. Adula in the diagram. In the diagram, எல்லா திக்கனிங்குமா சேர்த்து போட்டுருக்கான். ஒன்றில் இந்த ஆனுலர் பிளேயராக இருக்கு, பிட்டட் பிளேயராக இருக்கு. மீதியெல்லாம் அந்த மாதிரி திக்கனிங் வரும். I hope you understand. Then see that xylem trachea, it is long, narrow, whereas vessel இந்த மாதிரி இருக்கு. சரியா? Then again, this tracheates and vessels thickening, and the thickening சொன்னீங்க. So it will be pitted thickening. Here there will be few, whereas in vessels there are many pitted thickening. அப்படின்னா என்னது? பெரிய pipeline it is not empty. It is filled with the thickening of the cell wall material. At the same time, இங்கே ஒரு plate மாதிரி இருக்கும். பிளேட்னா என்ன சொல்லுவோம்? அகெயின் ஒரு மல்டி ஸ்டோரி பில்டிங்கில் பைப்லைன் இருக்கும்ல, அப்போ ஒரு தேர்ட் ஃப்ளோர்லேருந்து செகண்ட் ஃப்ளோர் வரும்போது ஸ்ட்ரெயிட்டாக வச்சுட்டு ஒரு கட் போடுறது இல்லை, இந்த கட் ஸ்ட்ரெயிட்டாக இருக்காது, இந்த மாதிரி ஒரு கிராஸ் பண்ணிப்பா. அதுதான் இந்த மாதிரி we do like this. இதில் there will be one pore. There will be one pore, that is called perforation. There will be one pore, that is called perforation, that is known as simple perforation. When there are many, it is known as multiple perforations. I hope you understand. Then the function of xylem is to conduct water and minerals from the soil by the root hair, and it is only unidirection, that also upward direction. I hope you are clear. The next one is phloem. Phloem is conducting food material as phloem. It will be having as sieve tube. The components, the tissues are cells. Phloem is formed of sieve tube, companion cell, sieve tube, companion cells, phloem parenchyma, phloem fiber. This phloem fiber is called bast fibers. Even the parenchyma. Okay, so now this sieve tube, it will be having structure like this. Even the sieve tube. And then every sieve tube will have companion cell. Every sieve tube will have a companion cell. Sieve na chalade chalona. So when the food materials are transported from the leaf to the different parts of the body, movement of molecule will be by selectively permeable or semi-permeable chalona. So that is controlled by the sieve tube. So this is called sieve plate. This is known as sieve plate. Okay? And then this is sieve tube. And in flowering plant angiosperm, sieve tube will not have nucleus. It is a living tissue, but it will not have nucleus. Only companion cell will have the nucleus. Companion cell will be helpful for functioning of the sieve tube. Okay. Same way in xylem, tracheids will be present in gymnosperm. Gymnosperm non-flowering plant pteridophytes and gymnosperm. The tracheids will be present, vessels are absent. Whereas in angiosperm, tracheids and vessels will be present. Same way in phloem, companion cell is absent in pteridophytes and gymnosperm, whereas in angiosperm, companion cells are present. I hope you are able to follow this. Are you clear? Yeah? So simple, permanent and complex permanent tissue as xylem, tracheids, vessels, parenchyma. Xylem fiber is called wood fiber. Whereas phloem xylem 9 cells adhe madri idilayum 9 cells examples a group of tissue. Some will be differentiating into sieve tube, companion cell. The remaining are called parenchyma tissue, and this parenchyma tissue, when it becomes mature, this is called bast fibers, and they give mechanical support. Wood fiber and bast fiber will give mechanical support. I hope you understand. Tracheids will be present in lower group of plants as pteridophytes and gymnosperm, whereas vessels are absent, whereas it is present in the angiosperm. Same way in phloem, companion cells are absent in pteridophytes and gymnosperm, present in angiosperm. Then appearance of this vascular tissue, first occurrence of vascular tissue, xylem and phloem started from the plant group pteridophytes only. So primitive type of occurrence are present from the pteridophytes. I hope you understand. So this is for the complex permanent tissues. I hope you understand. Okay. So this is about the xylem and phloem. Are you clear? Right. We will continue in the next session. Thank you.